It's a pleasure talking with you today on a topic that I really find close to my heart, and that's genetics and neurogenetics. What we're really going to concentrate on today is some basic concepts and what clinicians need to know about genetics, genetic testing, and some neurogenetic disorders. I hope you find that the talk today enjoyable, and I want to thank you for the hour of your time that you devote to listening to this talk. Our goals today are to understand the basics of human genetics. These are the things you need to know to get through the day and get through your clinic. We're really not going to be talking about any specific human diseases. We want to talk also about recognizing the need for genetic testing in sick children. It used to be that genetic testing was incredibly expensive. And just to give you an idea, that when the human genome was first sequenced, it was a project that cost about $100 million to sequence one person's genome. When it first became available on the commercial level, about 2007, it costs about $150,000 to sequence your genome. And the data was delivered on actually a MacBook. When it was introduced as a clinical tool in 2013, the cost was $16,000. And now the cost is clearly under $1,000 to sequence the entire genome. And I think in the next few years, we're going to be talking about the cost in terms of about $200. And really, in the end, for any child, you really don't know what's going on with their illness. I think very soon we're going to be talking about getting a CBC, a CMP, a lead level, and an exome sequence. And I know that sounds a bit over the top, but I think we're getting close to that. I also want you to understand the value and limitations of basic testing and some concepts that you need to know before the geneticist arrives. I'm going to start with some basic concepts of DNA, messenger RNA, and proteins. The DNA double-stranded molecule is incredibly stable, and in fact, our DNA remains stable for decades after it leaves our body. You can lick a postage stamp back in the days when you could lick postage stamps or the back of an envelope, and your DNA will be stable and ready for sequencing even 50 years later, as long as it's stored in a cool, dry place. The messenger RNA is actually a negative of the DNA. It's coded in an opposite direction of DNA and is used by the body to make proteins. Messenger RNA is very unstable and starts to decay within minutes of it being formed. So whereas the DNA in our nucleus is there, for lifetime, and can replicate and can make messenger RNA. The messenger RNA has a very limited lifespan within the body or outside the body. And this is why the messenger RNA vaccines have to be stored at super cold temperatures and must be given within really minutes after being constituted and brought to room temperature. The messenger RNA contains the code, and we'll talk a bit about the code in a few minutes, but contains the code for the structural and enzyme proteins within our body. And this is just a picture of the enzyme that converts methemoglobin to hemoglobin. And I just chose it randomly as a picture of an enzyme protein. So let's talk a little bit about the human genome. Again, it's a double-stranded DNA. It contains 3.3 billion bases, and because they're paired, there's actually 6.6 billion base pairs in the human body, and contains in this 3.3 billion as a primary sequence about 20,000 genes. So it takes 20,000 genes to constitute a human, and that's an approximate number, and it's still in somewhat of a debate whether it's 16,000, 18,000, 20,000, but for argument's sake, we'll just call it 20,000 genes. As it ends up, only 1.5% of this genetic code is actually turned into messenger RNA. And this is called the coding region. It also is called the exons. And again, the coding region makes up the messenger RNA of our body. which again ends up being translated into proteins. So the first question one would ask is, well, if it's only 1.5%, why have the other 98.5%, which is non-coding? These are also referred as to the introns or intronic regions. As it ends up, this used to be called the dark matter, almost as if it had no function. But now we're learning that much of this non-coding region has other critical functions in the body. There's very little difference in the actual coding regions between humans and some other mammals, but the humans end up having a lot more non-coding regions. So somehow these non-coding regions are affecting the function of the coding regions. This would include regions that serve as promoter to actually let the body know when that gene is supposed to be turned on and when it's supposed to be turned off. And this non-coding region can form microRNAs that regulate gene expression in other ways, and one other method is called RNA silencing. For the most part, what we're still dealing with in 2021 is the coding region. And when we order a whole exome sequence, what we're really doing is ordering this 1.5% of the total DNA. There is a test on the market called the whole genome sequence test, and this is starting to explore that other 98.5%, although we still don't know what a lot of this region does and how it affects human health, although we know it does affect human health. Aside for sex differences, 99.9% of our DNA is exactly the same as those sitting next to us, regardless of race, heritage, or other distinguishing factors. Ultimately, our genes are turned into proteins. The genes which are part of the DNA get transcribed by a particular enzyme into messenger RNA. The messenger RNA gets translated into proteins on a structure in the cell called the ribosome. So just to give you an example of what these proteins can look like, on the left there's the protein dystrophin, and dystrophin is a muscle protein that when mutated, when the gene is mutated, can result in Duchenne's or Becker's muscular dystrophy. On the right is the protein called the transmembrane conductance regulator protein, which when mutated can result in cystic fibrosis. So this is an illustration, or two illustrations, of the molecule of DNA. And some people refer to it as a twisted ladder, as you can see with the illustration on the right. And a closer view of this twisted ladder will show several components. The green pentagon is a ribose, and actually in DNA it's deoxyribose, and that's part of the backbone. The blue ring right here indicates a phosphate group, and the different shaped bases represent the nucleotides adenine, thymidine, cytosine, and guanine, A, T, C, and G. And these A, T, C, and Gs, there's only 4 different molecules that form the base linkage that hold the twisted ladder together. And you'll notice that A always pairs with T, and C always pairs with G. And this is what we inherit from our parents. And again, just a simple view of what a molecule of DNA looks like. There are some differences between DNA and RNA. Again, going over this, DNA contains the sugar deoxyribose, whereas RNA contains the sugar ribose. DNA is double-stranded, RNA is single-stranded. DNA is very stable, RNA is not stable. DNA is responsible for storing and transferring genetic information, while RNA is the negative blueprint of DNA and directly codes for the amino acids and acts as the messenger between DNA and the ribosomes make the proteins. And the DNA and RNA base pairing is slightly different. As I said before, DNA uses the bases adenine, thymidine, cytosine, and guanine, whereas in RNA, uracil is substituted for thymine. And uracil differs from thymine in that it lacks a methyl group. At this point, the clarification, you can sort of put that in the back of your mind, because really what we're going to be talking about is the sequence of DNA or the sequence of RNA, not why we have uracil on RNA and thymine on DNA. Again, DNA is made up of a sugar, deoxyribose, not ribose, a phosphate group, and 4 basic molecules. And again, these molecules, these bases, are arranged in groups of 3, and they're read in groups of 3. And when we think about it, those groups of 3 could be CGG, ATG, TGA. If I wrote them all out, there would be 64 different combinations that are possible. And of course, they're not 64 amino acids which are coded for, and the amino acids are what strung together make the proteins. There's 21 amino acids that are coded for, and so there's some repeat in the genetic code, and I'll show you a slide that will show you those repeats. There's certain sequences that actually tell the ribosome, stop, we're done with making the protein. And these are called stop codons. And of course, you want your stop codon in the right place. And any mutation that results in a stop codon in the wrong place will deliver a shortened protein and therefore a non-functional protein. So these molecules are strung together in a huge strand that compose the molecule called DNA. And only one strand of the DNA is duplicated to make additional copies. And only one strand is transcribed into RNA, and this strand is called the sense strand. The other strand is the antisense strand. And the other strand, again, in DNA is there just to protect the molecule and keeping it as a stable molecule. And this AT and CG pairing are constant throughout all life on Earth. So this is the slide I was promising you. And again, this is the genetic code as it would appear on the RNA molecule, because RNA has a uridine or uracil and not a thymidine. So when the ribosome reads UUU or UUC, it knows that the corresponding amino acid to put on the protein is a phenylalanine. Likewise, UUA or UUG would result in a leucine to be put on. There's actually 4 more combinations that code for leucine, and this is a very interesting AUG, which codes for methionine, or an initiation codon, which is the cue for the ribosome to say, hey, let's start making a protein. And then you'll see UAA, UAG, and UGA are the stop codons, which again is the signal for the ribosome to say, stop making the protein, we're done. It is absolutely not important to memorize any of the order on these on this slide, just to understand the concept that 3 bases in a row give the ribosome the cue as to what amino acid to insert next on the protein, to start the protein formation or to stop the protein formation. And again, here are the amino acids, the building blocks of proteins. And again, just to, you've probably all heard about these amino acids at some point. When strung together, these amino acids can form the protein insulin, can form the protein hemoglobin, and can form any of the 1,000 different proteins. Obviously, different orders and different lengths of the proteins result in very different functions of proteins. This is another cartoon representation of what all this would look like. DNA is stored in the nucleus. It is unraveled and transcribed into messenger RNA. This messenger RNA leaves the nucleus into the cytoplasm, enters a part of the cell called the ribosome. The ribosome is what translates the triad base of messenger RNA into a corresponding amino acid, and these 5 dots here represent the starting formation of a protein, with this dot here being the first amino acid, second amino acid, third amino acid, fourth amino acid, fifth amino acid. And as the ribosome marches down the messenger RNA, transfer RNAs attached again to the triad and add on the next amino acid in the sequence of the protein. So let's talk a little bit about mutations. Now, mutations occur when the sequence of the DNA is changed, and of course, if the sequence of the DNA is changed, the sequence of the messenger RNA is changed. So the A, T, C, or G is changed to another nucleotide. There are other types of mutations than just a switch. You can actually have an insertion of a nucleotide, which then puts the corresponding subsequent nucleotides out of order. And again, the genetic code will get all messed up when that happens. And so your leader part of the protein may be perfectly well formed, but because you insert an A or a T or a C or a G where it doesn't belong, the triad sequence that follows is nonsense information and will make a totally non-functional protein. And that's called an insertion. You can also have a deletion, and you could drop one of these bases. And again, all subsequent triads will be out of sequence and then form a nonsense protein. You can also have duplication of one or more nucleotides. And so you may have an insertion or a duplication of 1, 2, 3, 4, 50,000, 100,000 pieces of genetic material. Big duplications can occur. Trisomy 21 is an extra copy. It's a major duplication of a chromosome. You can also have missing chunks of DNA, and this is called a large-scale deletion. And these are referred to as copy number variant disorders. So when you see 16p deletion, that's a copy number variant disorder, or 22q duplication, that's a copy number variant disorder. And there could be there's a numbering system that could, come up with 22. Let me start again. There's a numbering system following the P or Q, which designates the short arm or the long arm of the chromosome, such as 22q11.2 deletion results in something called the George syndrome. And that 11.2 specifies where on the chromosome is that either missing or duplicated piece of information. Mutations have no moral. Some mutations are advantageous to the individual or species fitness. Some mutations are disadvantageous to the individual or species fitness, and these cause diseases that we see all the time. And other mutations are totally neutral and appear over time and don't have any particular benefit or disadvantage. Mutations typically are inherited. That's how we think about them, but they can occur spontaneously, after conception. Spontaneous mutations can occur when they occur pre-conception. They're called germline mutations. When they occur post-conception, they're referred to as somatic mutations. When we're referring to any one particular point on the human chromosome or the human genome, um, or one particular base that is typical, we refer to that as the wild form. So if in 99.999% of people there's an A at that particular point, A is referred to as wild, where T, C, and G would be referred to as mutant. And there's another interesting nomenclature. If the mutant is actually non-disease forming or non-disease causing and is present in a large enough percent of the population, like 1%, it can be referred to as a polymorphism, which is just a non-pathogenic variant that occurs in a large enough population but isn't typical. So it isn't, the wild type. And so we like to be able to classify mutations as wild or mutant. And for the purpose of the next few seconds of the discussion, we'll refer to mutant as disease causing. So because we inherit 2 copies of every gene and 2 copies of every nucleotide at a particular position, you could have homozygous wild, homozygous mutant, or heterozygous. And the heterozygote state is when you have 1 copy that's wild and 1 copy that's mutant. So we have 2 copies of every gene. The main exception is respect to males and the X chromosome. For most genes, 1 perfect copy is adequate. These tend to be genes that encode for enzyme proteins. And with enzyme proteins, if you have 50% normal amount of that enzyme, you'll live a healthy life. A disease occurs when you have 2 bad copies, and these are referred to as autosomal recessive inheritance disorders. In general, one parent will have, or both parents will have mutated copies unless the spontaneous mutations occurred, and there's a 25% risk of recurrence with each pregnancy. Examples of autosomal recessive disorders are sickle cell anemia and cystic fibrosis. For other genes, you need 2 perfect copies to have a healthy existence. These tend to represent genes that form structural proteins in the body, and diseases can occur when you have 1 mutated copy. In general, one of the parents will have a mutated copy and be symptomatic as well, although spontaneous mutations occur, and you can have a situation where neither parent has the disorder, but the child actually has the disorder. A great example of this is neurofibromatosis type 1. In most cases of NF1, you have 1 parent that has NF1 as well, but in 20 or 30% of circumstances, neither parent has NF1, and the child has developed this as a result of a spontaneous mutation. Huntington's disease is another example of a disorder that's autosomal dominant in terms of inheritance. So we talked about these points a bit ago, but I'm going to go over them again to drive home the 2 major types of mutations that are important for most genetic disorders. The 2 major types of mutation are either a point mutation or a copy number variant that results in a deletion or duplication of genetic material. So a point mutation is when the A or the T or the C or the G is changed to another letter. Another type of point mutation is when a single letter is dropped from the sequence. This will result in an amino acid change or a change in the amino acid sequence of the protein encoded by the gene. It can also result in the occurrence of a stop codon resulted in a shortened protein. These shortened proteins are recognized by the body as not supposed to be there and are deproteinized and destroyed almost immediately. So a short protein doesn't do you any good, and tends to be a very serious, the stop codons are a very serious type of mutation. And just as an example, sickle cell anemia is a disease caused by a point mutation in the beta globulin chain of hemoglobin, causing a an amino acid, glutamic acid, which is hydrophilic, to be replaced by the hydrophobic amino acid valine in the 6th position. So essentially all sickle cell anemia is caused by this one specific point mutation. Deletion or duplication is when a sequence of alleles, and this sequence can be a couple hundred, couple thousand, 20,000, 200,000, or a whole chromosome, is deleted or duplicated. And when we're talking about deletion, it's no big deal if the other copy doesn't contain a gene with autosomal dominant inheritance characteristics. But it is a big deal if it does. So it gets, in trisomy 21, we're talking about an entire gain of function of an entire extra chromosome. In velocardiofacial syndrome or DeGeorge syndrome, we're talking about a deletion that spans about 30 different genes. And so the other copy of the gene ends up not being enough to make up for the difference in loss of genetic material. And this is an old visualization of what these copy number variant reports would look like when you can see a loss or gain in genetic material. So let's talk about some different types of specific mutations. A missense mutation occurs when there's a replacement of a single nucleotide. You can see how this genetic code, CAT, gets converted to a histidine, CAT, another histidine, CAT, another histidine. And here, if you replace the A with a C, instead of a histidine being here, you have a proline inserted. And this may produce a malfunctioned protein. And of course, malfunctioned proteins can lead to disease. Nonsense mutations in general result in a stop codon, which terminates mRNA production, resulting in a short non-functional protein. So in this sequence, instead of the CGA, which corresponds to an insertion of an arginine, you get UGA that creates a stop protein or stop codon. And so instead of having this normal amino acid sequence, you get the stop here and a shortened protein. Again, disease causing. And finally, deletions are another mechanism of genetic disease. If you're missing all this genetic material, you can have disease result because you're just missing a whole bunch of genes. 2/3 of Duchenne's muscular dystrophy are caused by deletions. And we all have heard of Cru-du-Chat, where the short arm of chromosome 5 has been deleted. I'm not going to go too much into methylation. Only want to mention it because when you hear about terms like epigenetics or environmental causes that relate to diseases like cancer, often it's a discussion about methylation. And what methylation is the ability of the body to turn on and off genes in an inappropriate fashion. And we're going to be hearing more and more about this as time marches on, and as we get more information out of the whole genome studies that we're now starting to order, as opposed to the whole exome studies. So there's two main types of genetic testing that we can do. One is testing for point mutations, and the other is testing for copy number variants. Although interestingly enough, with the newest technology of whole genome testing, when you start testing for one type of mutation, you'll start testing for two types of mutation. At the beginning of the talk, I mentioned that the cost of genetic testing has dropped dramatically. And this is a graph representation, and if you I want you to follow the green line, is the cost of the first human genome was $100 million, And again, the cost when this became a commercially available for a non-medical purposes, it was, wealthy people wanted to know what their genome showed, not that it was well interpreted. It was about 2007, 2008. You could buy a sequence for about $150,000. This became commercially available in 2013, and here it shows the price of around $10,000. The first the first ones that we were ordering were $16,000, and now the cost is settled. The commercial cost at about $1,000. The actual cost of doing this is debatable, but some people say it's as low as $200. And we're charged $1,000. Our patients are charged $1,000 because there's got to be some profit to be made for what used to be an incredibly expensive test. I can tell you, when I started ordering gene testing frequently, I was paying, and this was about 2007, I was paying about $2,000 per gene to get sequenced. Now we're talking about $1,000 for 20,000 genes. So the cost has dropped dramatically. Moore's law is a computer chip technology that basically calculated that the cost of storage, computer storage, dropped in half every 18 months. And so the cost of sequencing human genome has fallen more dramatically than the cost of computer storage. So again, as I mentioned, in 2021, when we order a test, we can test for point mutations or we can test for copy number variants, although it's all going to come to the same thing soon enough. So we can order a test for one specific nucleotide chain, testing for one gene. And this sort of gets to the point, if you're working in a pulmonology clinic and you're worried about cystic fibrosis, it makes sense to test for a single gene. I'm worried about CF, I'm going to do genetic tests for the gene that causes cystic fibrosis. And again, the commercial cost of single gene tests runs now between $500 and still about $2,000. If you're worried about alpha-1 antitrypsin deficiency in the pulmonology clinic, you can again test for that one gene. If you're working in, let's say, a mitochondrial clinic like I do, and I see a child with a disease that looks like Lee syndrome, and again, I'm not going to get into what Lee syndrome is about, it ends up there's about 75 different genes that can cause Lee syndrome. So companies have formed panels of genes that address one specific illness. Lee syndrome is a 75 gene panel. Epilepsy, they're panels that range between 250 and 500 genes that test for all the known genetic disorders that result in epilepsy. We can also order a whole genome test, and that tests for all known genes, or a whole genome test, which tests for all known genes, the areas of the intron that have been explored and that we understand. And this ends up, the whole genome technology has allowed us to detect deletions and duplications and additional genetic changes, like trinucleotide repeat disorders, which I haven't even mentioned. Now, let's just think about this conceptually. If you're going to, and let's just skip the first one, because back in the old days, genetic testing was so expensive, you sometimes actually had to pick what nucleotide you were looking for, because that's all they were going to look at. So, so back in the old days, and what I mean by old days is 10, 15 years ago, you had to have a hypothesis-driven question to order a genetic test. I'm looking for sickle cell anemia. I'm looking for cystic fibrosis. I'm looking for Duchenne's muscular dystrophy. I mean, I can tell you back in 1995, you couldn't test for any of these. Okay, so all this stuff sort of started happening around the turn of the century. But it had to be hypothesis driven because, again, early on, if a patient had ataxia, at the time there were 17 known genes that caused ataxia. You started hunting at $2,000 a pop for gene 1, gene 2, gene 3, gene 4. Now there's dozens of these genes that we know that result in ataxia. Back then you really had to have a very hypothesis-driven, hunch before you started fishing for different genes because it was just way too expensive. As we moved into panels, all you needed was the patient's got epilepsy, the patient's got ataxia, the patient's got pulmonary dysfunction. You can start ordering panels of genes, and now with the whole exome and even whole genome, you almost don't even need a hypothesis to what's going on with the patient. All you need to know is that the patient's sick, has a genetic disease. I don't mean to sound anti-intellectual, but when we get caught up in our busy clinics, we have to become efficient at what we do, and sometimes we don't have the 20 hours to investigate what's going on with a patient. I can give you a really good example of such a situation. There was a child in our neonatal intensive care unit several years ago who spent weeks and weeks with test after test showing different things wrong with the child, and really no one was able to put it together. Very smart doctors, but no one was able to put it together. A lot was resting on knowing what was going on because the parents were really debating about what to do with this child's medical care, whether to do nutritional feeding or not, withdrawal care. It was really complicated. I went and saw the child and realized I was up to my eyeballs in complications, and I made the decision to order a gene test, the whole exome test, which came back a week later, gave us the diagnosis and total clarity on how to treat the child. And the child's doing very well now. Not a healthy child, but we knew exactly how to treat and how to counsel the parents, and they were thrilled. They thought I was great. All I did was order a test. And I think we're seeing whole exome come into the clinic to help solve these problems. Now, it's not going to solve every problem. And when I do talk like this, my geneticist friends sometimes do get upset because they really feel you do need a hypothesis before you start ordering genetic tests, and I don't disagree with them. But when you've had dozens of doctors see a child and not know what's going on, and then the genetic test solves it in a week at a cost of $1,000, I think we need to be practical about this. There's also a test called a microarray, a chromosomal microarray, which is a very efficient test for copy number variants, looks for deletions and duplications. And again, whole genome is going to be replacing this chromosomal microarray in the next few years. We also have karyotype, which is an older version of copy number variants, and there's still a purpose for karyotypes on occasion, but for the most part, chromosomal microarrays have replaced the karyotype, and pretty soon whole genome testing is going to replace it all. And again, I don't mean to be anti-intellectual, but if you get a whole genome at a cost of $1,000, we charge $4,200 for a sedated MRI scan. The economics sort of weigh heavily towards whole genome testing when it comes into play. And what this slide predicts is that pretty soon, and it may not be 2022, but maybe 2025, is that genetic testing may be replaced by whole genome testing. that will be very cost effective at looking at everything genetically. Now, having said all this, we're very lucky at Akron Children's Hospital to now have a robust genetic service with lots of providers and genetic counselors that actually not only take care of us at Akron Children's Hospital, but the entire region, including adult hospitals. So not all hospitals are have this type of genetic service that we have here at Akron Children's Hospital. And again, our genetic counselors serve as cancer geneticists for the adult population in the city of Akron and surrounding areas. So we've got a lot to be proud of. And really, at first iteration, if you've got a patient that you suspect may have a genetic disorder, certainly consulting genetics is the way to go. Depending on your particular type of practice and how comfortable that you feel, I believe that there are responsible ways for APPs and physicians to order genetic testing. I haven't gotten into the informed consent portion of any of this, which is I think the hurdle before anyone actually starts ordering genetic testing. There's a lot of stuff that can pop up on a genetic test that ends up being particularly upsetting to families, not just learning about the disease that they may have or not have, but diseases that they weren't even suspecting. So when ordering a whole exome test, as an example, you're looking at BRCA1. You're looking at genes that predispose to horrible diseases like breast cancer. And so we had a situation in my practice where I was looking for a neurogenetic disorder, which we weren't able to find by the testing, but what we found is that the child was a carrier for BRCA1. And as a boy, that does have some relevance, but not as much relevance as we later found out that his mother and sister also had that mutation. They didn't know about it. Now, you could say, well, it's a good thing they found out, but it's a better thing that we told them about that this test could result in them finding out about diseases or disease predisposition that they haven't even thought about. Genetic testing can result in finding out about non-paternity, that the biological father isn't who the family thought was the father. Again, consanguinity can also easily pop up on genetic testing. So this is not a talk to let you know to start ordering genetic tests, but I do want to drive home the point that I believe sincerely that we are ordering too few genetic tests at our hospital. The American Academy of Pediatrics has come out, and other organizations have also endorsed the recommendation that any child with developmental delays and dysmorphism undergo genetic testing. That's just as a start. Any child with multiple dysmorphic features probably deserves consideration for genetic testing. And again, the pathway for genetic testing, if you don't feel comfortable or you're not qualified to do informed consent, is through our genetic service at Akron Children's. I hope this talk has given you a better idea about the human genome, how genes are arranged on the human chromosome, the difference between DNA, RNA, transcription, translation, protein formation, the different types of mutations that can occur, and the different ways that we can explore the human genome. There are projects going on in other children's hospitals where all sick neonates are getting rapid exome testing to try to uncover the the cause of their illness. I think we're getting pretty close to that ourselves, and hopefully in a couple years we'll really be in a very nice situation. We'll be able to help children and their families with rapid diagnosis. Thank you for your attention, and if you've got any questions, you can contact me by email, and be happy to discuss this further with you. Thank you.